It's a pleasure to be here, obviously, and all words about this splendid facility, I think, were said, and I don't need to repeat that. It's the second time I'm in this building, and it's impressive. I wish all our seminars are carried out in these rooms. When I was preparing this lecture, I was thinking about the challenge that I had. So what was the goal? To entertain you, but I'm not a comedian, I cannot really make it very funny. Or to teach you, but you are not students. Or to show off myself, but again, I'm not an artist. So it's difficult to make it so that I can satisfy fully myself and I can satisfy everyone here. I'll try my best. And I think the goals that I have today is, first of all, is to help you understand a little bit about what cancer is, how cancer develops, what is the future of cancer medicine that we see today, and a little bit about how science, our science at York, contributes to all that, and that's probably it. And I would like to make it interactive. If you have questions, if there is a misunderstanding, even though you don't have a microphone, and your voice would not be recorded this way, but please do not hesitate to ask questions to make sure that what I talk about is as clear as possible. And let me start with the slides. One thing that I've done is I changed the title a little bit. And the reason is that since I suggested the title, some things developed in Canada, and I was thinking about this development for quite a while, and this affected my today's lecture. And this new development is the Canadian government, the federal government investment that was made in a form of money and obviously in a form of will towards the development of so-called personalized medicine or personalized healthcare. And I just copied from the website of the Canadian Institute of Health Research a few lines that were in the minister's presentation on January 31st this year when this new initiative was announced. So it was suggested that the new healthcare that we are looking forward to will not be using the old approach where one single approach works for everyone. The new approach would be personalized, meaning that we will be tuning diagnosis and treatments based on individual characteristics of people. And this approach will benefit a number of different diseases in different disciplines, and cancer was listed on the top, oncology was listed on the top, but many other disorders were also suggested as the ones that would benefit from personalized medicine. There are some things that probably would not benefit that much, but in particular, cardiovascular diseases and neurodegenerative disorders such as Alzheimer's and Parkinson's disease and diabetes and pain management, all this will benefit from very careful individual by individual approach to medicine. And there was even an example given in that speech, and the example was exactly about cancer. And the reason for this is that today we foresee the best how personalized medicine will benefit cancer for one simple reason. Cancer is probably the most complex of those diseases because it's associated with mutations in the genome. Some of them are random, and because of this, it's impossible, theoretically impossible, to predict what would happen in cancer. The point is, and this is a message that I want you to take home, really. If it's maybe not a single one. I would have a few simple ones, but this is one. There are no two identical cancers. So cancer in one patient is different from another patient, and therefore, diagnosis should be individualized, personalized, and treatment should be personalized as well. So that's how the government wants us and what the government wants us to do. Now, the question that I have for myself, is it really the beginning or the continuation? And you all know that doctors look at us individually. So medicine is to some extent personalized. We go to a doctor not as a group, and the doctors look at us not as a group, but as an individual. But there is obviously a difference that the government had in mind when they suggested that there would be a future of this medicine, personalized medicine. So what does the government and what do we have in mind when we talked about personalized cancer care and personalized medicine? Now, to understand this, we need to look at how things are done today and how they will be done tomorrow. So today, how do we diagnose cancer? So from time to time, obviously, there are screening procedures. For men over 50 years old, there is a procedure in which we screen blood for prostate-specific antigen for prostate cancer. There are imaging procedures for breast cancer for women after a certain age. But in many cases, cancer is first diagnosed because of pain or because of a bump somewhere in the body, which is a very rudimentary way, and often it's too late to really do any kind of intervention at that stage. So what do we do next in a conventional medicine? After we have cancer diagnosed, after that, a drug is prescribed. What are modern or yesterday and today drugs? We call them cytotoxic drugs. What are they? Poisons. What they do, they would enter cells that divide frequently. Cancer cells divide frequently. But they would also enter cells that divide normally. Hair, sperm, germ cells, skin, epithelial cells in our digestive system. And because of this, you know there are side effects of cancer treatment. As a matter of fact, those cytotoxic treatments, their therapeutic dose, the one that helps to cure cancer, is only about 5 times lower than the lethal dose. So if we overdose by a factor of 5 only, then the person will die. That's a fundamental challenge in medicine, because if we think about what happened next, we prescribe a drug, we administer a drug, and then we see that the tumor does not respond. So what does the doctor do? The doctor increases the dose. How much can we increase the dose? I told you, 5 times. After that, that's the end of the story. So we need to change this paradigm completely in order to become efficient. So on the right-hand side of this slide, I listed a vision of what personalized cancer care would be. Obviously, this may change because this is the future. And we are just trying to predict how it will work based on our current knowledge. That is limited. So, blood tests that we have today, imaging tests that we have today, will they die? No, they will still be here. Pain, we will not rely on pain. No, we will rely on pain. Obviously, there will be screening procedures based on the genomic information. We will learn how to sequence the genome, and after that, we will know who can potentially be susceptible to, for example, familial or genomic cancers. But there are also sporadic cancers. No one knows whether he or she can get a certain cancer. It happens just because of the environmental exposure or something else. So we still need to screen for those things, not by looking at the genome, but by looking at the person's current state, which is not written in the genomic information. So what will we be using for that? we will be using what is called molecular biomarkers of the disease. What are these? These are molecules such as proteins, such as RNA, DNA, that may be floating in our blood, or may be present, for example, in the main tumor that we are able to localize and take a small biopsy from the tumor. So what we will be doing after we do primary diagnosis and see cancer somewhere, we'll take a piece of this tumor and do very detailed molecular analysis, trying to see the signature molecules that would predict to us what cancer subtype we deal with. When we have the cancer subtype, we will be able to prescribe what we call targeted drugs. What do those targeted drugs do? They are not typical poisons, cytotoxic drugs. What they do, they are designed to find a molecule in a cancer cell by interacting with which we can change very subtle but very important interactions between molecules that will signal to cancer cells to die. And only cancer cells to die, not normal cells. Those new drugs are supposed to be, the therapeutic doses of them should be 1,000 times lower than the lethal ones. So if the drug doesn't work, we can first increase the level by a factor of 2 or 10, and the person would not see side effects. And we would not see side effects in people who will be treated for cancer. Today we see that by loss of hair and by many other side effects. So that's the future. So there will be a smart therapy, which is chosen for specific cancer. Then, after we prescribe this therapy and after we administer this, we will be following on a day-by-day or on a week-by-week basis, looking for those signatures, molecular signatures, and trying to see whether or not the therapy works. If it doesn't work, what can we do? We can modify the treatment. We will have potentially more than one drug for one subtype of cancer. And we can add what we call modulators, things that change the response of tumor to this drug to some extent. So that's the future of cancer treatment. And I have another slide there are those books that I like Computer for Dummies or Cancer for Dummies. So the next slide, I wouldn't call dummies, for amateurs. So how do we see futuristically what cancer treatment is going to be? Let's just go through that. It's a hypothetical scenario. So a person undergoes a regular blood test or a regular imaging test and diagnosed with, for example, breast cancer. What do we do next? We know that breast cancer there are 56, for example, or 60 subtypes of breast cancer. We know that already. Now we need to specifically find which one we have. For that, we have molecular diagnostic tools, very detailed, which can be done with the biopsies or with blood or with other fluids, for example. We do that and we say, okay, we have subtype 42. For this subtype, we have a drug. We start prescribing this drug, but before doing that, we can also look at the genomic information and see what the genes tell us. The genes can tell us whether or not the tumor will be responsive to the drug, or potentially will be resistant to this drug. If we predict that the tumor will be resistant, we change the therapy. And then, again, we will be screening the person during the treatment, trying to see how the organism responds. And after we cure cancer, hopefully, we will be also continuing this monitoring with the molecular biomarkers for years to go in order to foresee the relapse of the disease and to make some attempt to prevent it fast and in the early stage. That's how we see personalized medicine will develop. Now, where are we? What have been done and what needs to be done? The first part, obviously, is here. The number one is here. Regular stuff is done. But everything that starts from point 2 is not here. So we don't have those biomarkers, comprehensive biomarkers. Many of you heard about PSA. Why did we hear about this? Because it's the only one for prostate cancer, and prostate cancer kills every second man in the developed world. Some of you may have heard about HER2, for example, biomarker for breast cancer. Why do you know about that? That's the only one. There is nothing else. Many of you know what kind of treatment we have for those HER2 breast cancers. They are terribly expensive. We are talking about $100,000. How much do they help? They prolong life by about 6 to 8 months. So that's not that efficient. We do not have, so far, in this futuristic procedure, steps 2 to 7. So we need to work towards that. And this work will be long and will involve lots of resources. That's why the government starts investing into personalized cancer care. And now, for us, for amateurs in this business, from time to time we read something about that and get excited. And sometimes get overexcited. And I want to warn us not to get overexcited. This is my recent reading. After the government announced personalized medicine, on March 3, I'm reading an announcement that there is a company, a startup company, that made a genome sequencer that allows us to sequence a human genome during 1 day for about $1,000. What is genome sequencing? For those who didn't hear about that, so we have genes, and there are many of them. There are tens of thousands of them. Doctors use genetic diseases term for years, but in the past we were only looking for 1 or 2 genes that we knew. So what can happen now with those genome sequencers or readers of the genome? We can sequence or read the entire human genome. The entire human genome consists of 3 billion letters. It's a lot to read, but this company made a device that can read 3 billion letters, one very long word, or actually if we consider genes as words, then one very long sentence of several tens of thousands of words. Every word would be thousands of letters. So that can be done. Then in the May issue of Scientific American, I'm reading a candid conversation with the 2 inventors. And they are bragging that this will revolutionize the cancer care, specifically about cancer. And when I read this, I simply do not believe. They oversell this point. Of course, it's very useful to have this. But from the beginning, will it solve all the problems? It will not. Why? Because you know that tumors accumulate mutations. In tumors, cells have different genetic background. They cannot, those devices cannot sequence the genome of the individual cells. So we still have some kind of blueprint from the whole genome sequencing. Will it be useful? No doubts about that. Will it cancel all the effort of the Canadian government and everyone else involved in this enterprise of looking for personalized cancer medicine? No. For decades ahead, tens of thousands of researchers will be working on that. Every new invention, every new breakthrough will help, but there is no a quick fix. We all know that because battle with cancer started somewhere in the beginning of the 70s. Today, 40 years down the road, there is no a universal cure. There is no universal diagnosis approach. There is full understanding that there is no universal. It should be personalized. There where personalized medicine comes into the game. And now, what is needed for personalized cancer medicine? This slide is scientific. It's difficult to be entertained with this slide. So we'll go slowly and try to understand it. I said that we will have very detailed molecular diagnosis approaches. And for that, we need to know the molecules, the culprits of cancer, that the signatures of those molecules will be indicative of a certain subtype. But we still don't know those signatures. So our task number one is identifying molecular marker signatures that would be specific for certain cancer subtypes. And to do this, we need first to have tools, methods, instruments that allow us to do this kind of search. And I'm involved in this business. I know the state of this business. What I know is that $1 billion a year is spent for what is called biomarker discovery. How many biomarkers do we have in the field? We heard of HER2, we heard of PSA. That's it. We cannot validate them. We cannot start using them. Why? Validation of a signature of 6 biomarkers working together would require screening 10 million patients with a known outcome. So we would screen rather not patients, but bank tissues. And maybe people died already, maybe they're still alive. We need a medical record. We screen those tissues from a paraffin-embedded, for example, samples. And then trying to associate, correlate the signatures with the outcome, clinical outcome. And after that, we make conclusions about what signatures are real and what are not real. And in the previous slide where I showed to you those announcements, I didn't talk about the last one. I talked about 2, but the last one was in the Science issue, the latest that I got, and it was about a challenge to one of the biomarkers clinical studies at Duke University for one simple reason. People who do research do not know bioinformatics, or maybe play with bioinformatics to present their data in a way that would mislead the experts and the public at the end of the day. So bioinformatics is a huge issue here. We deal with tremendous volumes of data. How can we manage this? There should be algorithms, computer programs, super powerful computers to shuffle all this data and get the information out. Okay, so number one was finding signatures, molecular signatures. The second problem is to understand the molecular mechanism of cancer in order to start doing new drugs, smart drugs. What do we imagine as smart drugs? I told you. That would be molecules, potentially proteins, that will go through our body, find the cancer cell, and attach to a cancer cell. Then they will be internalized inside the cancer cell. And then they will find the molecule that is a key molecule, a therapeutic target, bind to this molecule, switch off its activity, and because of this, the cancer cell will feel very uncomfortable and undergo what we call apoptosis. Apoptosis is a programmed cell death. That's our imagination. But to make it work, we first need to have full picture of what molecules do. We call this the molecular mechanism of cancer. We know only a very, very rough blueprint of that, maybe 1%. I don't want to be over-optimistic here as well. This data have been accumulated over decades. We think that we know a lot. We know more than my or your brain could comprehend. But that may be only 1% of what we need to know, or maybe 0.01% of what we know, really. And the final thing is that with the genome, we need to learn how to read the genome. The second, we need to associate certain genomic patterns with the disease. And for that, we again need to screen millions of patients with the known clinical outcomes, and then use those complex algorithms that still don't exist and super power computers that may still not exist to digest the data and give us and doctors very clear protocols for how to deal with patients. So that would require decades of dedicated research of many people. So we understand that, and we are ready for that long marathon. Science differs from many other enterprises because science is a marathon run by sprinters. You need to run very fast, but for very long time. And what do we do? What does my lab do? We are technology people. We are developing technologies for that first task and the second task. So we are developing practical methods that would allow us to quantitatively measure multiple molecules in blood or saliva or urine, or very small samples from what doctors call fine needle aspiration biopsies. When they take a small sample from tumor, it may contain only 100 cells. And in order for us to get the molecular signature out of there, we need to have very sophisticated technologies. That cannot be done with lakmus paper. Right? Today, there is a tremendous effort in science, in some branches of science, to simplify diagnostics. We hear that. There are, again, a huge amount of money spent for that. It's good. That's good because finally some of diagnostic, very simple, can be brought to Africa, for example. But for complex diseases such as cancer, the lakmus paper will not work. That's another message that I would like you kind of to reflect on and to take with you. And today, time is limited, and I would be able to maybe touch a little bit. Only this part. We are developing highly sensitive tools, research tools, for understanding molecular mechanisms of cancer at the single cell level. So with this said, I need to explain you what the molecular mechanism of cancer is and why single cell level. So before I start talking about what we do, I still need to explain you a lot. And let's try to go through this as well. So what is the molecular mechanism of cancer? People say this and often feel what it is, but do not exactly know. Even researchers, you ask them and they would say, okay, let me think about this for a couple of minutes. Let me formulate that. You Google for that, you will not find a single definition. So we will derive a definition today for our internal use, the one that satisfies me and satisfies you as well. So to understand what the molecular mechanism of cancer is, we first need to understand what the normality is. Why are we having a certain shape and a certain size, and that doesn't change much? Of course, I can gain a few kilos, I can lose this, but in general, this shape, the Vitruvian Man and Venice, it's been done hundreds of thousands of years ago, it's still the same. So it's somewhere imprinted at us how many cells we need to have during our life, approximately. And then there is a balance between that. So the normality is that balance which is accomplished through a very fine interplay of three developmental processes. That's what biologists say, developmental processes. One of them is cell proliferation, multiplication, which goes through cell division. You have one cell, it divides into two. The second is that programmed cell death, or apoptosis. Programmed cell death, or apoptosis, is something that the cell cells do from time to time to make sure that there are not too many of them. So it's normal because we need cell renewal. So cells die and new will come in there and take their place. There is also one more developmental process which is not that intuitive. It's called differentiation. It's easy to understand. Cells, different cells in the body do completely different things. So they need to differentiate. Different creatures need to be formed from the same stem, original stem cell. So the interplay between cell birth, cell death, and cell specialization, so going through the university study, will be the normality, right? How does this interplay happen? How does this balance happen? This balance happens through an amazingly complex interplay of hundreds of thousands and millions of molecules. Very complex work. And researchers, when we look at this, we are trying to picturize that. We create what we call molecular interaction networks. One example of this is shown at the bottom right of this slide. It shows that some molecules, those little shapes are molecules. And somehow they interact. How they interact? They bind to each other sometimes. Sometimes they can react, and chemical, covalent changes can happen with them. And this is used to transfer signals. For example, if there is a hormone that comes to the surface of the cell, and the information needs to be passed to the nucleus where genes will react, there are so-called cascade of signal transduction. So molecules talk to each other. And this whisper goes from the surface of the cell inside the nucleus through a set of chemical interactions. So we build this kind of pictures to better understand what molecules do with each other in the balanced state. Now, what is cancer? Obviously, to understand cancer, we first need to understand normality. Because cancer is abnormality in comparison to normality. So cancer is an imbalance. And cancer is an imbalance that starts with the perturbations in molecular interaction networks. Those perturbations, we often don't know what molecules do not play well. But what we know that for some reason, proliferation takes over and goes faster than cell death. In an uncontrolled or poorly controlled fashion, the organism loses control of its own parts. And then what happens? Cells grow faster and tumors starts growing. What's the problem with tumors? They grow, let it grow. The problem is that tumors take place of normal tissues. And if instead of tumor cells in stomach do not digest food, that's the problem. There are many of them, but they do not do what they are supposed to do. They simply forgot the differentiation, the university education. They forgot that what they were taught to do. They became naive. They only know that they need to multiply. That's it. That's why we end up with death, because organs do not do their normal work anymore. They are replaced with non-specialized, undifferentiated cancer cells. So now I think we can make this definition of what cancer, molecular mechanism of cancer is for our internal use. And I define it as the interaction network in cancer cells, molecular interaction network, compare it to the normal ones. So if I see that in this complex network, this is a little bit a more complicated one than in the previous slide. You see that there are many more molecules interacting. And again, this is a small fraction, a minuscule fraction of what the full reality is, obviously. And if we see this in normal cells, and then in cancer cells, we see that there are a few distinct changes. We say the molecular mechanism of cancer is in those distinct changes, in the known pattern network of molecular interactions. Does this make sense? Good. I think that's another kind of home take, or how do you call this, take home message that I would like to leave with you, that cancer mechanisms are those molecular abnormalities that differ interaction networks, molecular interaction networks in cancer cells from normal cells. Now, we don't know much about molecular mechanisms of cancer. We need to learn a lot. So we need to learn that. That's the picture. And intuitively, we understand what we need to do. We need to take a cell and do extremely sophisticated analysis, chemical analysis of molecules. So we need to measure quantities of all those molecules, ideally. But in addition to that, we need to know how molecules interact with each other. We need to know reactions, real chemical reactions that happen inside the cell. Is it easy to do? No, tremendously difficult task. Do we have tools today? Unfortunately, we don't have tools for that. We're still working on developing very rudimentary tools for this. And there is one challenge that I haven't talked about yet, but the challenge that justifies the term single cells that I had in the title. We know that tumors accumulate mutations. They start with a mutation, and then the cells lose control of mutations because, first of all, they do not know how to fix DNA. Those genes that are responsible for fixing damages in DNA are lost. They don't work normally. And then their control over apoptosis is lost. Control over production, cell multiplication is lost. And from cell to cell, the cells divide, they accumulate those changes. We end up with something similar to the bottom right picture. This is a piece of tumor in obviously an artistic representation that shows that tumor consists of multiple clones of different cells. And even within a single clone, for example, those blue cells, we still expect to have cells which will differ in their genetic background and also in their phenotype, in the way what they look like. That's the ultimate challenge because, think about this. We need, first of all, to analyze and make some kind of sense of those differences between the cells. We already know, by the way, that the more differences we have, the poorer prognosis of cancer is. So we know that the cancers are more aggressive with greater level of differences. But in addition to that, there is a tremendous analytical challenge. If I want to understand why the cells in tumor differ and what level of difference we have, I need to have tools for that. I need to have an instrument that allows me to study chemical contents of individual cells to a great level of details. That's the challenge that we are going through. And there are some classical techniques, and you potentially saw some of the pictures. If you Google for fluorescence microscopy, for example, you will see those beautiful pictures. They are the most amazing. They're almost, for me, they're almost as amazing as the night sky with the millions of stars. Now, this is informative. Every color here corresponds to one type of a molecule. And we can, by distinguishing the colors, at the left-hand side, we have cells. The two pictures at the left are cells, where membranes are shown and the nucleus are shown, for example. And in the membranes, we stain with the color a certain type of molecules, and then in the nucleus something else. So with two colors, green and red, we can distinguish two types of molecules. And there is another technique which is called flow cytometry. It's almost like microscopy with the only difference that the cells do not stay long. They fly through a light beam. And we don't see the shape of the cell, but we record how much of light intensity of different colors is. And get something like the right-hand side picture, not much of information. Pay attention to colors. We have, in the right-hand side picture, we have 1, 2, 3, 4 or so. The point is that those classical techniques that we have rely solely on color distinction. And if you and I can distinguish lots of colors, those instruments, unfortunately, can distinguish up to maybe 10 colors. And for a 10-color instrument, you would need to pay $1 million. It's very expensive. And there is a limit again. Over 10 is impossible to do. So we need to create something else. And my lab is focusing on what we call chemical cytometry. So the previous techniques were image cytometry and flow cytometry. They're classical biological techniques, but they are made with no chemical analysis in mind, really. They were made by engineers and biologists. And we are saying, let's look at the problem as chemists. And as chemists, we need to do chemical analysis using what? Tools, sophisticated tools of instrumental analytical chemistry. So we designed this term, chemical cytometry. And now this term, I designed this term with my colleague, and we published the first article in 1999. In 2010, there was a book published with this term. I was not even invited to contribute. So the term lives on its own. It's like the kids, right? At 18, they leave the house, and they don't remember who gave them life. So this lives on its own. And there is a Canadian content into that, and a significant one. So in addition to my lab, that works with the optical detection, there is also a very successful story at the University of Toronto by one of my friends and colleagues, by Scott Tanner, who developed with his team, largely Russian for some reason. I don't know, Scott did not show to me any preference to Russians, but the team is Russian. And what they developed is an instrument called CyTOF, which relies on what is called mass spectrometry. Mass spectrometry is an expensive tool, of course, but it allows us to detect molecules based on their size and on their electrical charge. So every molecule, big molecule, has a certain electrical charge, or you can induce it. So the story at the University of Toronto, and now at a company called DVS Sciences, the story is great. They actually made an instrument. The bad side of that story, and I know that during those lectures, you are not supposed to hear bad things, right? So only pleasant stuff. But since we talk about cancer, cancer is not pleasant anyway, so I broke the rule. And I would say that York lost the chance to have this team at York. Scott Tanner was here at York and was talking to the Dean of Science at York, and at that time, the Dean of Science, or the administration, was not wise enough to give him a chance to be here. So we need to learn from that. And the other bad side of the story is that DVS Sciences is an American company. So Scott went around and was trying to find investors here. Investors here prefer to invest in gold and in oil, but not in that. So the company, and Scott is now, I just read that a few days ago, the company, the investors found a new CEO, a good businessman. Scott is not a businessman. He is a real scientist. A pleasure to talk to a person who would not twist your arms to get the last penny from you. So that's probably the reason. I haven't talked to him since January, so I don't know what the reason of replacing him, but probably there is a more business-oriented person who took leadership in the company. Now, our story. The mass spec story is great. And the mass spec story allows a lot of things to do, but it doesn't allow everything to do. What we are trying to develop is much more complicated and would not be done in a year or in two. I have a decade-long plan for developing that. And this story, how am I doing with time? Does anyone monitor me? Typically when I teach, the students, when it's time, they just leave without saying thank you or goodbye.
Stay with you.
So do I have five more minutes? 6 minutes? Okay. So what are we doing? we rely, this is a little bit of science. We rely on a technique called electrophoresis. Raise your hand if you heard the word electrophoresis. Raise your hand if you heard the word electrophoresis. From science or from a clinical setup, from physiotherapy? So what is electrophoresis? Molecules have charges. If you place them in an electrical field, then they will be moving with different velocities depending on the charge. This allows us to separate different molecules physically. So remember those classical stories that I showed, pictures with colorful cells. The problem there was that they relied only on color. We are saying that we are physicists, chemists. We can separate molecules, and by physical separation of them, we can distinguish many, not only 10, but maybe 100, maybe more than 100. If we also multiplex this or combine this with different colors, then we will have double gain. Multiple colors plus physical separation. So we rely in what we do on this technology, on electrophoresis, but we do this in capillaries. So those of you who heard electrophoresis and potentially have done this, you probably have done this on gels in a more classical way. We do this in capillaries. So what is a capillary? I have an example of a few capillaries, and these are not the thinnest one. I took the most visible ones. So I can take a look, just pass it around. You can open and take an example and look at this. Very, very low diameter, small diameter glass tubes. They are not very brittle. They're coated with some plastic outside, so you can even try to bend them. The outer diameter of a normal capillary we work with is the outer diameter of my hair, and the inner diameter may be 10 micrometers, 20 micrometers. You would not see it with your eye. So we run electrophoresis inside this capillary. The size, the diameter, the inner diameter of the capillary is the diameter of the cell. So what's the idea? Is to insert the cell inside the capillary, open the cell, lyse the membrane, and then apply an electric field and separate the components inside the capillary by electrophoresis. This is the basics of our chemical cytometry technique. So electrophoresis, in the way, schematically, the way how it works, we just have a capillary that you have in your hands now, and I have the capillary drawn here. This is my capillary. It's inserted into two very small reservoirs with the electrolyte, and there is a high voltage apparatus that supplies an electric field to the capillary. So when we apply this high voltage, then the molecules move from the left to the right along with the solution. There is a laser source of light that goes through the inner part of the capillary. And when the molecules pass through the beam, then we can detect it with a light detector. And what we see is a certain pattern where the x-axis, the horizontal axis, is time. And the y-axis is the intensity of light. So every peak, every mountain in this picture corresponds to one molecule that passed through the detector. So that's the basics of capillary electrophoresis. And the instrument that we currently have is not similar to the Scott Tanner's device. The Scott Tanner packaged this in that yellow box very nicely. We still have it on an optical bench, all open for researchers only. We do not invite guests, especially young ones who do not know when to stop themselves. And there is high voltage, 30 kilovolts that we play with. And basically what we do is we inject a cell inside the capillary, lyse the cell, and then separate the contents. How do we inject the cell? The trick is very simple. The cells are placed on a microscope slide. This is a microscope slide about 0.1 millimeter thick. So this thickness is 0.1 millimeter. And the cells are in their physiological solution on the top. We place the capillary, the one that you have looked at, vertically on the top. And under a microscope, we can see if the cell is superimposed with the inner bore of the capillary. So this is my capillary, this is the cell, in front of the inner bore of the capillary. Then I apply suction to the other end of the capillary. And the cell goes in. And this is the cell before I suck it in, and then when it's inside, it disappears because it's out of focus. And you know what focus is, right? When you take a picture of someone, if you take a picture of someone who is very close to you, you don't see what is far. So now the focus was on the beginning of the capillary, and the cell is deep inside, and I have a picture where the cell is. So when we inject the cell, it's about 0.3 millimeters from the end, and then we lyse the cell. Now the cell is gone, you see only some bright things that are distributed through the capillary, and we made them visible only for the purpose of visualization. And then we apply 20 to 30 kilovolts and separate the contents. That's the basic principle of this technique, and I think I need to now, or in 20 minutes or in 2 hours, I can talk forever about this. This is something, that's a no-end story, but I will be punished later on. So I need to wrap it up, and let me maybe show one slide with conclusions so that we do this smoothly and not just me being kicked out from the podium. So I wanted to brag a little bit. We are recognized not only by the community. There are lots of stories written about the lab in different media, in different scientific media about us. And there are some things that stick around. In addition to chemical cytometry, there is also kinetic capillary electrophoresis, which was called by someone a Swiss Army knife in analytical chemistry. So we've developed a number of things. And we publish a lot. This is a list of peer-reviewed publications from the lab, starting from January 1st last year to April this year. So it's 14 months, 25 publications. Some of them are in very high-profile journals in the field. The articles are not so interesting for the general public, but we still, being researchers, we still brag. We still like showing that we are productive in what we are doing. And the conclusions, my conclusions are very simple. So, how long will it take to develop personalized cancer medicine? It will take time. I wish that everyone here is still around when this is developed, and I wish all of you, long years, many years of active light, productive light with this. Then I wanted to also emphasize that modern science is very interdisciplinary. People work in different areas. All this comes as little streams come into big river. All this is mixed, and that's when we get the power. And in my group, I'm trying to make even my own group very interdisciplinary. I'm cross-appointed into physics, chemistry, and biology programs, so I can drain graduate students from 3 departments, which is a benefit. My PhD degree, Robert, is in physics. So I work in the chemistry department. I deal with biology. So that's the story. And I think it's beneficial for the students that come from different groups, from different backgrounds, to talk to each other. They become more productive, and the results are visible. Another thing that I wanted to say, that today, in research, in science, 90% of good work, discoveries, is done by very young people, by students, by sometimes undergraduate students. We are very proud of them, and that's what makes our life as professors so interesting, because we are always surrounded. If I don't look at the mirror, I still think that I'm 22. So, because we're surrounded by young people. And every project in science leads me to the same simple conclusion, that science is always wrong. Never solves a problem without creating 10 more problems. But those 10 more problems is exactly what we will be doing tomorrow and the day after tomorrow. That's why it's self-perpetuating forever, from day 0 to day infinity. And at the very end, I would like to obviously acknowledge people who make my life so interesting. It's my team. It's my young friends, my students, who are doing 99% of work. And I typically have a pleasure of only presenting this stuff, maybe helping to digest it a little bit. So this is my team. That would be impossible for us to do the discoveries without generous support of the government. And there are a number of government agencies that support us. And I know that researchers often complain when they say, well, the government wants us to do something. They announce a certain program. Why don't we have an academic freedom to work to do what we want to do? I don't agree with that. The way how I think about this is that 100 years ago, the governments did not invest in science. Scientists would need to go and find philanthropists and ask for money somewhere else, and someone else would give them the money. Today, the government gives the money. The government gives the taxpayers money. The government wants us to be accountable and to do something that the taxpayers would like us to do. So that's normal business in my view, and I'm very happy that we were successful with all this stuff. Collaborators. Without multiple collaborators, it would not be impossible, even though we are very interdisciplinary. We don't know everything. We cannot do everything. That's why people from Oxford University, from the University of Toronto, from McGill collaborate with us, or we collaborate with them to get something interesting and exciting. And at the very end, I would like to really thank you very much for surviving this torture. And, if I'm invited next time, I would not be so brutal. I would talk either about politics in Quebec. It will be done today, so I need to invent something new. Thank you very much.